an honour to be here, an honour to be part of this series, both at FENS and as part of the consciousness series that Johan has been hosting for a long time now. As Johan was mentioning, this morning we had a short symposium, and I gave an introductory talk for this talk tonight. So I'll go very quickly over the first part of this, because what I want to talk about is the latest things that we've been finding in a obscure, very understudied layer of the cortex. Let me just get this right, which is referred to as layer 6B, or sometimes layer 7. Cahal referred to this layer as layer 7, but these days it's generally referred to as layer 6B, and it's a very thin part of a thin layer of cells right at the bottom. But it makes no sense to start talking about layer 6B without a bit of introduction as to why it would be interesting and how it could possibly be related in any way at all to consciousness. So I'm going to start by talking about dendritic integration theory, and just to prime you on this, if you haven't seen this kind of an image before, then you're probably struggling to recognize it. As soon as you see what the original image was, when you go back to this image, you'll see it immediately, and this is a well-known case of priming. It also has the property that if you see it again now, even years later, you'll recognize the frog straight away. And that's a testament not just to the way that the cortex works in terms of priming, that's to say your expectations drive what you actually perceive, but also this goes very quickly into memory. I won't talk about that side of it. This morning I was talking about what we think is the memory aspect of this. In any case, this idea that you're using, essentially you're predicting what it is that you perceive, and those are the things that you tend to see, has been, I think, framed very well by Anil Seth, who's trying to get us out of thinking of perception as a feed-forward process, but rather a feedback process in something that he calls a controlled hallucination. But he's, of course, coming from a very top-down way of looking at this, and my heritage is doing single-cell recordings in slices. I started in the laboratory of Bert Sackmann in Heidelberg back in the 90s, recording directly from the dendrites of cortical neurons, and there we wanted to know what the biophysics of these neurons were. It's been a long arc to get to where we are now. Of course, these days we're mostly looking at these cells in vivo, and I'm going to try to convince you that we can go from the biophysics all the way to this kind of statement. The key finding back in the 90s was what we call backpropagation activated calcium spike firing. And what you're looking at here is the result. The black trace is the recording from the cell body of a layer 5 pyramidal neuron from the cerebral cortex, actually a rat at the time, and this is the big layer 5 neurons that you can see here in profile. This is from a mouse, but they look almost identical. And the red recording is from the dendrite of the same neuron at the same time. And once you've got a recording like this, you can play God and inject whatever currents you like. And here, enough current is being injected at the cell body to cause the action potential every sweep. And then there's a simulated compound EPSP being injected into the apical dendrite. And if I stop this at the right place, well, first of all, the way to, I think, look at a pyramidal neuron is that it's got two major arborizations that are receiving inputs. They're not so easy to see the bottom here, but you can easily see the tough dendrites. And essentially, what's going on here is that at particular timing of the action potential that's propagating actively back into the dendritic tree, there's the collision with the input coming into the dendrite, which is activating a new kind of current, a calcium, the high voltage activated long-lasting calcium current that makes this plateau potential, just like in heart muscle, in fact. And that takes over the neuron, and the neuron bursts, and it turns this neuron into an associative device that's basically detecting when input is coming to both ends of the cortex at the same time. So the next question you should have is, what's the input that's coming to the top and the bottom? So I'm going to do this in sensory cortex, which I now think, after all these years, is a let's say, the wrong kind of priming, the bias, the terminology that we use. But nevertheless, for the purpose of this, I'll take the perception in terms of low-level features of an object, and I'm using a tiger as the demonstration. And what we know is that the cerebral cortex processes the different features of whatever objects you're looking at in really different regions. And in human cortex, they're centimeters apart. They're really very different. So color, orientation of the stripes, the movement and form, they're all being processed in very different areas of the cortex. But what we know is that the different areas of the cortex have more or less exactly the same architecture. They've got the same kinds of pyramidal neurons and presumably are doing very similar operations. And in point of fact, we're talking about columns of neurons that all have these large layer 5 pyramidal neurons. In fact, they all have all the other types of pyramidal neurons as well. But focusing on these pyramidal neurons, for each feature, what we would expect is that at least in primary sensory cortex, that the information coming from the eyes through the lateral geniculate and so on will affect the bottom of these neurons predominantly. And this has also been shown in current source density recordings, showing where the current sink is in the first instance. And of course that's bound to get some kind of firing in these neurons according to the biophysics that we see in vitro. But at the same time, there'll be lots of input coming from lots of other areas that are impinging through the horizontally running fibers in layer 1 on the tough dendrites. And at that point, one would expect that a certain subset of these neurons, namely the ones that are getting input in both directions, will fire these dendritic spikes, and that they will then start massively firing, which will, of course, send more feed-forward input, and then there'll be more feedback input, and the prediction is that you'll fall into a low energy state, effectively, where just the right ensemble of neurons are firing with no orchestrator or let's say homunculus needing to determine which ones should fire. These are just the ones that both receive some feed-forward data and some expectation at the same time. All right, I'm going really fast here because I said all this morning, and I guess that I'm boring half of the audience. But just to say that we have predictions on the various levels of this question. And coming from a bottom-up perspective, this is really important because it means that we can ask questions that we can... This basically translates into predictions of what we should see at the cellular level and in various conditions. And one of the ones that we tested like this was the perceptual threshold, which, again, I showed this morning. But just very quickly, we had a hypothesis that if in a famous kind of but very low-level behavioral paradigm where you simply test when either a subject, it could be a human subject or an animal, says that they see the stimulus or feel the stimulus. In the case of an animal, what that means is you train the animal with a robust stimulus to respond for a reward. And so now you can go through the different stimuli in terms of intensity, and you can see when the animal starts to lick for the reward, and that becomes its report of the the stimulus. So it's a fairly easy thing to test, but nevertheless, if you do this in a human, you can also ask the human, did it feel like something? And in humans, at least, it does feel like something at the moment of perception. It's not just that it's not an automatic response in a human. Of course, we can't ask the mouse what it's actually feeling here, but we, at least for the time being, assume that it also has a subjective experience at this point. Okay, so we're asking what happens at this point, which is really a matter of definition. This is, by definition, the point at which the person or the animal 50% of the time perceives that intensity of stimulus, or 50% of the time doesn't. And we're basically predicting that, or we predicted at that point that when you're in this quadrant, then there would be no calcium in the dendrites, and on the other hand, there'll be calcium in the dendrites above threshold. And well, to cut a long story short, this is the case. I don't want to dwell on this just because I want to get to LA6B, but as I say, I don't think you can really appreciate why we're excited about LA6B without knowing these facts. So the last thing about this particular study was that we could also manipulate the dendrites. And so here we had ways to specifically downregulate the activity of dendrites, and only in the cortical column that we were interested in, which for this task, which was a whisker stimulation task, was the C2 barrel. So we were basically only putting our, we did various different ways. We had light on the dendrites in the C2 barrel. We had a spritzing on a drug which caused an agonist for inhibition onto the dendrites that's particularly effective. And we also did the opposite, which was to express channel rhodopsin in these neurons and then stimulate the dendrites. So if we inhibited the dendrites, we moved the perceptual threshold to the right. That meant it was harder for the animal to perceive the stimuli. And if we put light on the dendrites, then we could move it to the left, so the animal we were assisting the animal in some sense. And in fact, we speculate that because the animal is still licking when there is no stimulus anymore, this is actually zero stimulus, that we're actually causing it to hallucinate in this case, which would perhaps be the controlled hallucination that Anil Seth is looking for. So that's all by way of preparing you for the 6B. There's one more finding that's important to go over, but just before I do that, hopefully you can recognize this now and you can do the test on yourself. I presume that you're all pleased to find that this kind of priming lasts some time. So I'll come back to timing and priming and so on from the LA6B, but for the time being, we want to put on these glasses of two-compartment neurons, which is really what's going on here. These are it's not just two compartments for the sake of two compartments, but two compartments in order to separate, in the first instance, two different kinds of information and apparently bring it together with a very nonlinear process. And I want to talk about anesthesia at this point too, because we chanced on a finding with anesthesia. So this was the point at which we thought that it was time to start talking about consciousness, because it was the flip side of anesthesia. But the result itself is really about anesthesia. So I had another very talented postdoc in the lab, Moritaka Suzuki, who was probing the questions that we were looking at with fiber optics and extracellular recordings. And what he was trying was expressing channel rhodopsin in these pyramidal neurons and then making a beam of light with what we call a periscope. It's actually an upside-down periscope, so a mirror, essentially, that's inserted into the top of the cortex and shines a beam of light sideways. And then you're in this fantastic position. You can finally ask the question, what happens when the distal dendrites of pyramidal neurons are active? And in the first instance, it was just what we expected from the in vitro, which made us happy, of course, because that meant that things were operating in vivo, like in vitro. What you're looking at there is a juxtacellular recording, so it's a kind of halfway house between extracellular and intracellular, It's actually extracellular, but it's attached to a neuron. So these are the spikes from from one pyramidal neuron. Moritaka Suzuki also did these experiments with extracellular electrodes, and then we could see the population response at the same time. And I'll show you a few of those. But what shocked us was when the same animal in exactly the same condition was anesthetized, this flatlined. And you might think at first, oh, that's not very interesting. It's anesthetized and everything's been downregulated. But the reason why this is shocking is that the channel rhodopsin isn't downregulated. So the thing that we're doing to the neuron stays static, and that was something that we could actually control for. So the anesthesia wasn't changing what the channel rhodopsin did. But nevertheless, it flatlined. And what it implies is that there's some really delicate mechanism or property across this axis that basically causes the anesthetic, for whatever reasons, to prevent the same depolarization that's occurring in the tuft to have no effect at all on the output of the neuron. And so, as you might think, this becomes then a hypothesis in itself. Is that the action of the anesthesia, or at least is that part of the action of anesthesia? And is this happening ubiquitously across the cortex? And, well, we tested 3 regions, the somatosensory region that we usually test in, but also prefrontal and visual cortex, and they all had this property. So we think that it's ubiquitous. Of course, there's a lot of work to go to prove that completely, but it seems that we're already hypothesizing that this is a across-the-board shutting down of the top of the pyramidal neurons, which is, in fact, a shutting down of all the layer 1 contextual input that would come to these neurons. And on more than that, which was I think, started for us, or at least for me, I think I was way behind the game on this, but we started thinking about thalamocortical loops already from some data that I didn't have a chance to show you, but I showed in the morning talk. But nevertheless, if you look at the anatomy of projections from higher order cortex to the cortex, you often see this pattern, depending on which part of the cortex you're looking, but it's very common to see a projection from higher order thalamus that goes both to layer 1 and to layer 5A, and people rarely talk about this connection to layer 5A and what it means, but it's actually right in the coupling zone of where these oblique dendrites are on the pyramidal neurons. And so it's perfectly situated to control the coupling at the same time. And sure enough, when we put an inhibitory drug into the thalamus, we actually got the same result in terms of coupling. So it's the same experiment. We got light on the distal dendrite, and essentially we were able to suppress. So now you're looking at the population response, and the control is the red one, and the purple one in the background is what happens when you block the thalamus. And so you could actually have the effect of an anesthetic by blocking the higher order thalamus, and at the same time this did nothing to the input to the distal dendrite. Mind you, the input at the dendrite is the channel rhodopsin, so that's not necessarily surprising. In addition to that, we found that various neuromodulators, and these are blocking the action of metabotropic glutamate receptors or blocking the action of metabotropic cholinergic receptors, had also this astonishing effect on the coupling across the neuron. In the meantime, that's been published. In the meantime, we've been looking at this in vitro, and essentially we've been able to show the same thing. You can really recreate this in vitro, which we're excited about because we can start testing this for various things. Of course, one thing you can't get in vitro is an awake slice. But what we did was to turn this upside down, assuming that it's not so much we assume that it's anesthetized, but that it doesn't have the input from the thalamus, and therefore it's presumably not coupled for that reason. But what we can do is now use an agonist for the same receptors that we used an antagonist for in vivo and show exactly the same effect. And I think that if we were barking up the wrong tree here, you wouldn't expect to see these effects recreated in a completely different preparation like this. So we actually think that this is something built into the operation of the pyramidal neuron. And what it tells us, I think, in the end, is that not only is there some cooperativity going on, there's not just an associative device, but there's a regulator on the association of the top to the bottom. And that's where I think layer 6b enters. But just to finish on this topic, we assume that consciousness has something to do with the thalamocortical loop. But there's been quite a lot of speculation as to whether the anesthetics are blocking or disturbing the thalamocortical loop, or whether it's really something to do with corticocortical feedback. And there's good evidence for both. And our claim would be that's because it is both. And it's both because if you block this delicate region here, then basically the lights go out, whether this is because this line is inhibited or whether this line is inhibited. And so the action of the anesthetic could be anywhere on this plot, and if it's going to affect this point of let's say it's a single. Although it's distributed across all the pyramidal neurons, it's essentially one mechanism in the end that would take out all the feedback, all the prediction that you would have. And so in as much as you need that prediction to perceive something, this should just take away that prediction you have for perceiving things. All right, good. And I'll just say that what we're expecting is that under normal circumstances you have completed thalamocortical loops, and you have a so-called integrated set of activated columns that are appropriately coding for whatever is the experience that you have, and that under anesthesia you're basically localizing this, and it can't spread out. Now, that's a quick summary of dendritic integration theory, which you can read about in the article that Johan was talking about. And I guess this is a very bottom-up way of looking at consciousness. And it's not the claim here that somehow that explains everything about consciousness. It's just the claim that these are observable features of consciousness, at least in mammals, and that there are testable facts that we can push at and ask now questions to do with consciousness in general, but also it provides us with lots of predictions. And I realize I'm going too slowly, so let me just go straight on to layer 6b or layer 7. So what is this? This is the thin layer at the bottom of the cortex that I was talking about. It's very much associated with arousal and wakefulness. It has so-called orexin hypocretin receptors, which means that it's very much bound to sleep-wake cycles. And orexin was found to excite these neurons at the bottom of the cortex. In fact, so I should say, what I'm showing now is all the work of a very gifted postdoc in my lab, Tim Zolnick, who would very much like to be starting a lab of his own now, so he's on the job market. Bad for us, good for him. I think anyone who would get him in their department should be overjoyed. So he one of the amazing things that he did was to go through all of these different receptor types that it turns out are part of layer 6b. So these cells at the bottom of the cortex, basically have every neuromodulation known to man. What I was showing you in the first slide was a dopaminergic expression of a construct that was activated by dopamine, which is just one of the things on this list. But basically, everything that you can imagine, it's being expressed in these cells, and one way or another, some of them excite and some of them inhibit. And so that's one reason I think this is a really important layer of the cortex. Another reason is it's very special. It's the only layer of the cortex that doesn't get thalamic input. So it gets long-range and local cortical input, but it doesn't get any thalamic input, be that first order or second order or higher order thalamic input. There's just, when I say it doesn't get any, the trace amounts of some thalamic input, but nothing like what the other layers get one way or another from the different parts of the thalamus. On the other hand, the neurons of layer 6b have a really special projection target, and that is you'll see that layer 1 and layer 5a here. And if I compare that now with, this is our favorite higher order thalamic region of somatosensory, the posterior medial nucleus, but basically that's the kind of projection pattern that you get from higher order thalamus. And you'll see here, it's not just got the same projection, but it's the same sub-projection. It's the same sub-layer of layer 1 and layer 5, which I think is a smoking gun. This and you can see that more forcefully here. So whatever it's doing is going straight to those nodes of the thalamocortical loop that we think are important for consciousness. And so this is seen side by side. This is the projection to layer 6b. So it's in the sense that this is exactly like the thalamic projection, then layer 6b is the cortex's thalamus. It's the part of the cortex that the cortex can drive, like higher order thalamus. And lo and behold, 6b also projects to higher order thalamus. So it's on every part, every node, if you like, of the thalamocortical loop, not just the same neurons to the same higher order thalamic neurons, but the same two nodes of those neurons in the cortex. So it's really got the thalamocortical loop by the scruff of the neck, and it's able to impose its way. I don't have time to go into the details here, but this is really to show that there's more than one type of layer 6b neuron. It gets a bit confusing at this point. They have slightly different projections. There's one type of them goes to layer 1, another type goes to layer 5a and to the higher order thalamus, and another type goes to the superior colliculus, which I think is also important. And basically, we think that this is all to do with regulating the thalamocortical loops, and therefore all to do with attention in the end, in one way or another. Tim looked at these connections really carefully, and you can see this is so. There's two papers. I think I went too quickly over that. The first paper is basically all the inputs to layer 6b, and the second paper is all the output of layer 6b, and they're really detailed and very hard to swallow papers, but are remarkable just in how much how methodical that he worked and was able to show what. So I think we really understand this layer at this point, and I won't go into the details anymore. But this is one of the things that he was able to show. So it's not just that they're going to the same layers that the higher order thalamus is going to. But when he looked at this, and basically this is a single trial in vitro, he's got, I forgot to mention that this all took off basically because we now have transgenic animals so Cree mice for two types of layer 6b neurons. And we have both of them in the lab, and they're not quite identical. They both hit slightly different populations, but they're also overlapping. Anyway, I won't go into the details of the transgenics, but just to say that we could express channel rhodopsin in the layer 6b neurons. This is the name of the transgenic, and there in the name you can see that this is taking advantage of the dopamine expression, dopamine receptor expression in these neurons. And so in these experiments in vitro, Tim did a patch clamp recording of the cell bodies of layer 5 neurons, both the ET and IT, that's to say the corticocortical neurons and the subcortically projecting big pyramidal neurons of layer 5. And he recorded from the cell body while he did a similar depolarization, but now it's not of the neuron, but of the axons of the layer 6B. So he's basically asking the question, what is the axonal input to this neuron, and what effect does it have? Well, it's astonishing. So what you're seeing here, this has been cut off. These are the action potentials that are cut off, but it's literally making dendritic spikes in the tough dendrites due to the light. This is several traces imposed on one another. And when you put in a blocker, a drug that blocks the NMDA receptors, you can see that these are actually NMDA spikes going on in the dendrites. And it basically flatlines. That's not exactly a flat line. What you see there is the effect of AMPA responses in the same dendrites. So you can see the difference between getting the nonlinearities in the dendrites or not getting the nonlinearities in dendrites. Of course, this might be partly calcium spiking as well, because we know from other experiments that I haven't had time to show that if you can get NMDA spikes in the local tough branches, then this will in itself cause calcium spikes, and so there's a cascade, if you like, of nonlinear effects that recruit the neuron. In any case, there's some really nonlinear things being driven by these neurons in layer 1. And you can compare that to what the higher order thalamus does, which is also not bad. It's not as strong as what layer 6B can manage, but it's nevertheless, it can be effective, can cause NMDA spikes in the dendrites, which again is shown by blocking NMDA receptors. And although we're showing it here for contrast, this is not a discovery of ours. This has already been shown in other laboratories. We could do this both for the ET stands for extratelencephalic, and these are the subcortically projecting pyramidal neurons. The IT stands for intratelencephalic, and these are the corticocortical ones. And the IT ones tend to lie higher in layer 5, and in particular in layer 5A, and so these are sometimes called layer 5A, and these are sometimes called layer 5B cells because they're tend to be in the lower layers and project subcortically. And you can just read off from here that the higher order thalamus has a larger effect on the corticocortical layer 5 cells than layer 6B. But basically, both do both. And the biggest effect seems to be in these big layer 5 cells, the layer 5B or the subcortically projecting cells. And so essentially what's possible for these neurons is to both target this region for the nonlinearities in this cell, but also this region for regulating the coupling from top to bottom. And of course, these are different populations of these neurons too, and I suppose over the next years we'll be trying to find out which ones of these are working under which conditions. But right at the moment, we are turning them on all together. Actually, Tim's found ways to selectively express channel rhodopsin in these different populations, so things will be moving on that time. And so just to pause a little bit here, if you imagine that we're now playing with some set of neurons, and I'm taking an analogy now to a Christmas tree with lots of neurons. You could imagine what we don't know, but we need to know at the moment is whether or not exactly how layer 6B is operating. Whether that means that it's coordinating in oscillations or the firing of neurons in a very coordinated way, or whether it's somehow selecting which neurons should be firing in the column, or Tim's favorite, which would be choosing ensembles and selecting for particular groups of neurons, and we just don't know at this point, but all of these things would be possible. I should say the speculation has begun at this point in the talk, and so I'm going to go through what we don't know and what we'd like to know, but what we can guess will be interesting consequences. So in any case, with regard to the various different things that we think that Layer 6b might be doing, attention is at the top of our list. But we know from work that came out of the Peter Jonas lab, which is in Vienna, that they have shown that Layer 6b has a very strong role in learning. We've not yet been able to show this, not because we just haven't got to these experiments yet, but we would guess that this is likely given the amount of non-linearity it causes. For reasons I guess I didn't have time to go into, we suspect that an important part of memory consolidation, in fact, in declarative memory, is in layer 1 across the board in the cortex. But, well, we might be able to get into that later. And of course, perception and arousal, wakefulness, and so on. So it's also all over the cortex. So essentially what you've got is a band which is, as I was saying before, the cortex's thalamus that is everywhere ubiquitously across the cortex. And that includes prefrontal cortex and primary sensory cortex, but also it's going into the parahippocampal structures as well. And so it's in itself a ubiquitous layer. By the way, I also failed to point out that these are originally subplate neurons that go very quickly into position. And the other thing that I didn't say, which is extremely remarkable, I think, and you can pick up in the first paper, the Cell Reports paper of 2020, Zolnik et al., is that it only contacts the layer 5 the layer 5 pyramidal neurons, IT and ET. It misses the layer 2-3 neurons, and which is quite astonishing. The layer 2-3 neurons have really extended tough dendrites all through layer 1. So it's some kind of feat to miss this, and there's really no input to it. And I guess that this is a developmental feature, that these neurons are coming in an early stage of development, and the layer 2-3 are coming in later. And for whatever reason, the synapse formation has finished from these neurons to the tufts. I don't know that that's the reason, but that's my guess at the moment as to how this is the case. In any case, what this sets up, I think, is the possibility then that you've got a cortical way to selectively unify across the cortex. And so this becomes a perfect way then to be a so-called conductor of attention. There's some sort of process that the cortex is in control of that can direct which corticus or thalamocortical loops should be engaged. So we did a simple experiment for the efferent study, which involved taking sleep-deprived animals that would go into in and out of deep sleep, and we recorded from every layer of the cortex with a multi-electrode array, and at the same time expressed channel rhodopsin in the layer 6b neurons, so that we could put light onto the cortical column and see what happens basically when you activate layer 6b in an animal that's not aroused. And essentially you see the evidence of slow wave sleep, and then the blue light here, which you can see coming on at intervals in this animal, sends this cortical column from the slow wave delta pattern into a high gamma. And this lasts just for the period of the light. You'll notice that the animal actually whisks after the light, so this is something else we want to understand. But locally in the cortex where we're putting on the light, it's all over at the end of the light. If you would do this if you would wake up an animal such that it went from delta to high gamma, it would never just shut down inside a few milliseconds and go back to delta again. This is a process. But so something is happening that is extraordinary in this condition. And in fact, it was also, you could measure this at a distance as well. So it wasn't just the local columns. So these are presumably also because it's being transmitted via layer 6b. I won't dwell on that. So now the other thing that's really fascinating is that it's not just that it has the higher order thalamic connectivity, and it's not just that it has the extraordinary powerful drive on the dendrites, but it also facilitates in comparison to the thalamic connections. So it turns out that the thalamus that's also driving the thalamocortical loop has depressing synapses onto the nodes of this loop, which means that, I guess, you're always fighting, particularly this time of day, and listening to a dense talk, to keep those thalamocortical loops going. And on the other hand, the layer 6b has a facilitating synapse. So I can show that in more detail here. This is the effect, so this you've already seen. This is the effect now of doing a train of impulses, light pulses, at the cell body of both the subcortically projecting and the corticocortically projecting layer 5 cells, and it's in vitro, so this light is put on the light's actually going on the layer 5A projection. And you can see the depression in both of these responses from the thalamic input. And if you do this with the 6B input, you can see actually to the IT neurons it's very facilitating, but also to the ET neurons it's still facilitating. And it's not just that it has this facilitating effect at these extraordinary synapses, but the facilitation facilitates or augments. So if you do blocks of these impulses, and you do these at quite long intervals beforehand, then you get a progressive, the dark blue is the original one repeated. And so these are, the purple ones in the background are the actual augmented responses that are happening over minutes and hours. So there's a very, very long potentiation of these inputs. We don't know how long it lasts at this point. But basically, we speculate that this is not just for controlling attention, but is potentially a form of short-term memory or working memory. So if you can imagine that you're using this process to control your attention, it's very... so the kind of overlapping concepts, the idea of working memory and sustained attention. But working memory, I think, would fit very nicely into this concept where for some time you actually are not using 6B, but the synapses know that they were used, and the next time they're used, they're ready to go even more strongly. That would be a kind of working memory, we guess. So now this sets up the same issue that I just went through at the level of a cortical column in terms of several columns. So we can ask the same questions. Is this a way to get synchronicity from one column to another? Is this alternatively a way to designate which columns are working? Or is it a way to get ensemble? And you can imagine that these will be, the answers to these questions will be important for various questions like perceptual binding and various other cognitive properties that we just don't know the answer to. But essentially, if you imagine, one way of construing this would be if you focus on here, you can choose to focus on blue or red or the fact that it's a ball. And in some sense, if you were choosing to do that, then you'd be choosing to pick one, or in principle, you could choose this by directing layer 6B to activate the right corticothalamic loops. And now I want to get back to the neuromodulation of this, because what this means is that so on the one hand, we're talking here about the selection of a possible selection of various loops, which we don't understand, but must be somehow related to what 6B is doing. And at the other hand, it's the hub for neuromodulation at the same time. And for all of the important neuromodulation that you can imagine. And so what we are proposing is that the neuromodulation acts such that if it goes above threshold, it starts doing its selection. If it's below that threshold, then it's not doing that selection. And of course, you could imagine everything in between. And so there's a regulation of the regulator here. And now you get all of those questions all together, but with a regulation of the temperature, if you like, of that process, whatever processes are going on, that you would have essentially the ability to control your level of, or ability to incorporate all of these. So at that point, well, anything's possible. So this is, of course, so the speculative part of this is that we don't know yet which of these modes it operates in and when it's most important. But basically, I think we can be quite sure that something of one of these trees is reality, if you like. So there's a few other predictions going on. We actually don't need, this is not speculation, so orexin, well, it's partly speculation, but orexin sensitivity or insensitivity leads to narcolepsy. It's quite possible that it's due to layer 6B, which has many orexin receptors. It doesn't have to be, because orexin receptors are found elsewhere in the sleep-wake cycles. But it will be as affected as other regions of the sleep-wake cycle, and it stands to reason that it would lead to momentary loss of consciousness, as happens in narcolepsy. It's also, we would predict, going to be important in various kinds of disturbances of both bottom-up and top-down attention disorders. And one of those is ADHD, which is known to be linked to dopamine and orexin. These are ways that you can actually treat ADHD, and in this case, this will both have an effect in terms of reducing the recruitment of the thalamocortical loop. But we also would anticipate that because it would tend to drive the higher order thalamus into burst firing, you would actually be distracted by the bottom-up features at the same time that you couldn't control the top-down features. It was well-known symptom of ADHD. So essentially, the disorder would be the losing this part of the loop, if you like, the controlled cortical control of this loop, and at the same time being actually too sensitive to bottom-up distractors. And at the same time, we would predict that there's a huge convergence of symptoms from schizophrenia to what could be all, I think, at least in part, if not in whole, dealt with by considering layer 6B. Again, it's known to be a disorder that's very much dependent on NMDA receptors and dopamine. And so it's quite, well, I'll go through a list of symptoms here. It's also related to poor working memory and the maintenance of attention, and lots of other lots of other, what at first glance seem like disparate symptoms that would come together, including the disposition to hallucinate, which would also be a factor of playing around with part of the loop going on here. Right in the beginning, I showed you the list of neuromodulation. It's complex because there's all of these different neuromodulators have different effects on different subsets of the neurons. So both excitatory and inhibitory, and there's 3 kinds of layer 6B neurons going on here. So although it's easy to see that anything's possible in some sense, finding what is actual will take some time. But in any case, we would offer it as a place that you should not overlook at this point in trying to come to terms with all of the things that are going on with layer 6B. All right, so the last thing I wanted to do is just throw out a few ideas that, because we do have some time to talk about these things. So this can be a discussion at large, but also I have to say that there were 2 fantastic talks from Björn Merker this morning and from Johan Storm, and we're talking about, I think we had some sort of convergence, and I think there is at least from my point of view, a kind of satisfying convergence of the different kinds of theories of consciousness. I guess this depends on your perspective, but the article that Johan is first author on, that I showed you at the beginning, and that has multiple authors who all subscribe to different theories of consciousness. We found a way, I think, to find a kind of cohesive way to look at all of the different theories of consciousness and find a through line. And so on the one hand, I would say that dendritic integration theory is not offering what we all want, which is why does consciousness feel like something? What's the where's the subjectivity? And basically the hard problem of consciousness is still eluding us, and I don't think it's even on the horizon using dendritic integration theory. On the other hand, what dendritic integration theory offers, I think, better than any other theory at the moment, is really predictions, and 6B is an example of one of those predictions. So when Tim walked into my office many years ago now and said, I've been using this transgenic and I can see the activation of these neurons at the bottom of the cortex, I said, please, I've been waiting for this day because I know it had been shown already that they projected to layer 1. The layer 5A projection was unknown at that point, and I basically could predict, using what we knew from dendritic integration theory, that this was going to be somehow interesting. I had no idea it would be as interesting as it turned out to be. But in any case, what we have is some sort of bottom-up set of claims that one can use for generating the next experiment. Just going through this, I think what we do agree on now is that it makes sense that the thalamocortical loop is really intrinsic for consciousness, and I don't think any of the major theories of consciousness would disagree with this fact, whether they be talking about integration or about the way that a workspace could have some sort of coordination and ignition time or level, whether it would be focusing on top-down input, which also is coming through thalamus, and that's giving a kind of feedback, top-down input to the cortex. So that seems, I think, to be a through line through all of the theories of consciousness. And one of the things that is missing, I think, from many of the theories of consciousness is a discussion of what's going on at the subcortical level. And I think that's what Björn Merker has been trying to tell us for decades at this point. And here, it would take me too long to summarize all of this contribution, but the point would be that the claim would be, no, it's not a cortical phenomenon. There's obviously, in mammals, which have a very developed cortex, that's adding some kind of richness to consciousness. But basically, if the subcortical structures are not involved, then you also are not conscious. So it's a one has to, I think, any theory of consciousness, and in mammals in particular, has to take account of both of these structures and their interaction and the way this works. And so, from that point of view, I feel like we're moving in the right direction, at least, and that we have a framework, a scaffolding, if you like, to bootstrap the next set of experiments. And there's all sorts of, we can do many of these experiments now in vitro, despite the fact that it's not conscious. We can test pathways and so on, and much of the work that I was showing you is done in vitro, but predicted things in vivo that are also proving interesting. So I put a few sort of starter questions at the bottom. Björn likes to talk about a pivot point, because I think on the one hand, I've been focusing on the distributed nature of cortex and how layer 6B might be, let's say, coordinating the distribution of activity. But this, I think, raises the next problem, that you don't actually experience this distributed nature of consciousness. It feels like a holistic thing. You feel like you've got one thought, not many thoughts. And so somehow this reduces down to a unified, integrated phenomenon from the first-person perspective. And Björn, I think, toyed with the idea that it might actually be the superior colliculus for some time, which is doing the coordinating point. And I think the motivation, if I can put words in your mouth here, is that basically, in some sense, it's, if you go to the mode of control, and this will be from basically all species that have coordinated in the world, they have to know basically what belongs to them and what doesn't belong to them. And so any individual organism has to have some way to access what belongs to the organism. And so there's a good argument, I think, that superior colliculus is involved in this. I won't. You can give your opinion on this as soon as we start the discussion. But I think this is a good starting point, at least this consideration that this has to be, that there must be some, what you could call this pivot point, that basically at some point owns everything that is a coordinated movement. And then you could imagine that this, if you don't have a cortex, that this is a set of structures that are phylogenetically quite old. But if you do have a cortex, interestingly, you've now got a tug of war, if you like, of what you want to do with your, let's say, directed thoughts, the nuanced thoughts that we think only the cortex can generate, and it has to be somehow integrating with the subcortical thoughts. So I guess that what we would claim with dendritic integration theory is that there's what the Germans would call a Knotenpunkt, which is on all of the important perceptory neurons of the cortex. And it's not so much that there is consciousness, it's that that's linking the distributed activity of the cortex into the subcortical milieu that is in fact directing consciousness in some sense. So it basically includes, that's the way to include the disparate or distributed activity of the cortex into what's going on. So I see I've got 45 seconds left. I'll just say that these could be starting points for a discussion or whatever you otherwise want to think of. This might look a bit obscure because I had to fit it on one slide, but the perturbation complexity index is a well-known now way to investigate the level of consciousness, particularly in people with locked-in syndrome, but also under anesthesia and under various different conditions. It's done with transcranial magnetic stimulation, and is basically testing how much a local stimulus of the cortex diversifies or extends into the rest of the cortex. And essentially the level of consciousness that we can deduce from other factors, like using anesthesia or locked-in people and so on, corresponds exquisitely well to how much complexity of the response this generates in the cortex. And so I think it becomes now a prime question as to what would happen if you interfere with 6B, whether that will interfere with these processes or whether they go on fine, and 6B is kind of peripheral to that. But there's a whole lot of stuff going on there. It just falls to me to thank all the people involved. I have a wonderful laboratory. A lot of them have, of the people, the data that I showed that's been going over many years now is done by people who've found their own way to different laboratories and different places. Tim, as I say, is still in my lab, but on the job market. But all of these people were incredible researchers, and I am lucky enough to continue to have a gifted team. So thanks very much for your attention.
Thank you very much, Matthew. Also very thorough talk. And yes, suppose there are questions. So Björn will go around with Björn, you will go around with the microphone. And also have questions.
Thank you. Okay. Björn.
Hey, it's a very beautiful talking, very interesting.
Thanks.
What I'm interested in is the relationship or interaction between dopamine cell and 6B RCB cells in the attention and working memory. Can you talk about the more detail about your idea about the interaction between these two systems?
If I got that right, you're asking what can I go into more detail about what orexin does? Is that right?
I'm interested in interaction between dopamine cell and the cortex cell in the attention and working memory.
The interaction between the cortical cells and I'm struggling to understand.
I mean,
Dopamine.
the mechanism
dopamine, yes.
how the dopamine cell and the cortex cell, the L6B cell, play a role in attention and the working memory.
So dopamine activates these neurons, and these neurons activate the thalamocortical loop. So in the case of dopamine, I think it's, well, except that there are subtypes of dopamine receptors and so on. So there are possible complexities there. But to a first approximation, we simply assume that activating the layer 6B neurons is activating attention. But as I say, at the moment, all of our claims are about 6B, quote unquote, but we know that 6B is different types of neurons. There are pyramidal shaped neurons there and non-pyramidal shaped neurons, and they're different projections. And so I can't tell you exactly in fine detail, I wish I could, which neurons are going to do what in under dopamine.
In addition or working memory process, dopamine cell and L6B neuron play together as a gating, or actually who getting who.
Yeah, of course they're not the only. So layer 5B neurons also also express dopamine. So it's not the claim that we can ignore everything else going on here. But since when we measure connectivity in the cortex in slices, the layer 6B by far is having the just wild effects. So if it comes to a competition between, let's say, the layer 5 cells and the 6B neurons, I would expect the 6B neurons to win. But again, this is I just have to hold off from making a really firm prediction at this point, just because of the complexity of the question.
Okay, so one more question. Do you think the L6B neuron mechanism you talk about is a general phenomenon at all of the cortical area, for example, vision area, auditorium area, or somatosensory area, all same, or specific?
Yeah, thank you for that question. If you take nothing else away from everything I've been saying, the major claim is that there's a recurring circuit that explains to a large degree what the point of the cortex is, which is a strong claim. But on the other hand, I think that I feel bold enough at this point to say that first of all, the pyramidal shape is the way it is because it's separating at least, or probably at most as well, two distinct classes of information. Exactly what to call these distinct classes is hard, but basically it's the dichotomy is internal versus external. I think if you believe in the cortex being divided up into feature columns, where these can go from low-level sensory features to high-level, even, let's say, rule-based features of frontal cortex, that there'll be, I think one way to look at this is the external is the feature-based information and the internal is the context. That's the words that I think are easiest to use once you start migrating from sensory cortex, where in the past we would have said feed forward and feed back. So if you imagine now that the pyramidal neuron is what we predict is doing a kind of Bayesian operation. It's saying each of the subcortically projecting pyramidal neurons is doing a probabilistic estimation of the probability of that feature in your perceptual space, and the point of the input to the distal dendrite is that this is the conditional probability. So it's basically the probability of this feature given the context, and that's basically what your cortex does as a whole. It does the probability of all these features given the context. It's just that they're all interconnected, and context for one neuron is the feature of another neuron, and so on. So this means that you can do a parallelized computation of the probability of all the features in your space given the context. And of course the context is changing all the time. The context is also changing because of what you're thinking. So this is not a simple calculation that's going on, but it's being done in a massively parallel way. So that's point one at the start. So I am getting to your question. This would be a principle, if you like, of the way that the whole cortex operates. And does 6B play into this operation principle? Yes, we find 6B in every region of the cortex with the same kind of connectivity. And it's jumping on top of exactly the nodes of this principle and basically controlling, either activating the context stream or fine-tuning the amount that context stream contributes to the ongoing probabilistic calculation of what what features should be prominent in your attention, let's say.
Thank you very much.
This has sparked all kinds of ideas in my mind. Some of the slides I eliminated this morning because of time constraints. One of them addresses your allusion to memory. Years ago, I was completely surprised to read a report from V2. This is early visual. This is the second step after V1. Mm-hmm. And V2 is sort of like a V1. It's just as big as V1. So it's early vision. They had a way to gene regulation manipulate layer 6B to enhance object recognition memory.
6B.
Layer 6B of V2 manipulated by this gene regulation. There's some complicated name, I don't know the molecular structure of it. Enhances object recognition memory. And then they went in and did cytotoxins and eliminated 6B and devastated object recognition memory. It blew my mind. How can V2 6B? But it fits with what you are saying very well. I can't remember the name of the authors. They are Latin American names.
I need to know this.
And I have the reference in my... So
that's fascinating. On that topic, so what I didn't talk about was the memory part, although I covered that somewhat this morning. And the connection from 6B to layer 1 was a smoking gun for us, also with relation to memory, because we know that we can block the parahippocampal input to layer 1 and cause devastating effects on memory consolidation. And stepping back from these questions, the importance, I think, of where we are now, which I keep trying to emphasize, but I try to reframe this in a different way each time, that there's no way to construe those results without a framework of what's connected to what and why should it matter. And so we happen to have gone the bottom-up way to these questions, but I think you can easily approach them from top down now if you have a mind now for these dynamics and keep that in mind. So to a first approximation, the hard part is disentangling, of course, all the multiple overlapping possibilities when you see it from a bottom-up perspective. But you have no chance at all, as far as I can see, if you don't know about these features.
Thank you for your lecture. And so every time I wonder what's the point of the difference with consciousness, lose consciousness under anesthesia. So because it looks like very similar the conscious lose at the anesthesia, but the consciousness is key point is so layer 6, but the anesthesia so is a key point is L5.
Yeah.
Is correct?
I wouldn't put it like that. Let me see if I can unwind that. I want to say that consciousness is not either of those things. The consciousness is some process that's integrating activity, not just within the cortex, but also subcortically, at a minimum. And that's as much as I can tell you about consciousness, unfortunately. So I don't feel like the expert on consciousness after having given a big lecture like this. All I can say is that we've seen some effects that relate to consciousness. But the two things you specified are doing, let's say, some things we would like to claim we know about now. So layer 5 is part of the important thalamocortical loop that is clearly necessary for consciousness. So I don't think that explains consciousness. It's just an observation that it's necessary. And layer 6B is impinging, or is if you like, the the thalamocortical loop supervenes on 6B, and 6B is playing puppet with the puppeteer, I should say, with the thalamocortical loop. Strangely enough, what that might mean is that 6B is your subconscious puppeteer in some sense. So one speculation that I didn't go into is that it's the gist part. So when you can't remember somebody's name, but you know everything about that person. You know their name starts with C and it sounds foreign and it's a long name and blah blah blah. You know everything about this name except what it is. One could imagine that your 6B is on the hunt and is activating thalamocortical loops, and the reason why you can't think about the name is because you haven't hit the right combination of thalamocortical loops that will eventually make you conscious, for whatever reason that I don't know, of the name. But in the meantime, you need some deliberate, some intentional way to search the space for this damn name that will eventually pop into your head. And when it does pop into your head, it'll be instant recognition in a kind of holistic way, and you can't go back. You can't go back to your former state, or at least not very quickly. You're then captured, if you like, by your own resonance of what's happening. That's one speculation that we have about what's going on. So I don't know if that answers your question. My answer is no and no, I guess, but yes, it's all involved.
Okay, thank you.
Okay. My turn to ask?
Yeah, go ahead.
Okay. Maybe I misunderstood something here, but I was wondering about what you said in the beginning about how the dendritic spikes might be involved in sort of predictions or integrating predictions somehow. But then we have the idea of, so if I understood correctly, the dendritic spikes are more sort of responding to the coincidence of the coincidence of a prediction and the primary input, sort of. But then there's the idea of predictive error coding that in some cases, or according to some models, the brain is focusing more on when the prediction is not matching the primary input.
Yes. Okay. Should I?
Yes.
Start
So
talking?
how does that fit in with that?
Right. Thank you. And so that's really well pointed out that this seems to contradict, I think, what is becoming, I think, a fairly well-established claim that not just the cortex but systems should operate according to predictive coding principles. And as you correctly stated, this is the inverse of this claim. So that this is the claim that if something is as you expected, it should stop firing, and that the things that should make things fire should be unexpected or should be error, as you said. Things that are incongruent with your expectations should cause a lot of firing. And at the same time, if you push people like Carl Friston to the point here and you say, well, is your prediction that the brain shuts down when something is congruent with your expectation? That's also not the claim. I think I think what evolution has done is that evolution is also conforming to the free energy principle here. And I'm not sure how well known fact, but it was surprising to me to find that when we started working in resected human tissue, you see this massive layer 2-3 in human tissue. It accounts for why the human cortex is another millimeter. It almost accounts for the reason why it's another millimeter thicker than the rodent cortex. There's a massive amount of layer 2-3 neurons. And what does layer 2-3 do? These are, I think, genuinely neurons that are behaving like one expects due to predictive coding. You could call them surprise neurons, or they're incongruent neurons. So the claim here, I think, would be, first of all, they do have a tuft in layer 1. All the layer 2-3 neurons sample the same, if you like, expectation line that's coming into layer 1. but they respond exactly oppositely, which isn't hard to with the same mechanism, isn't hard to arrange. You just need a negative sign at some point here, and you'll get exactly the upside down response. In any case, no matter how they do it, they do this, and on the other hand, they're very sparse firing neurons, much sparser firing than the layer 5. So my best guess as to what's going on here is that when you finally do see the tiger, and it is what you expected, this is not the time to shut down and do nothing. This is the time to react. But actually, you'd want to use as few neurons as possible for the match, if you like. And to the extent that it's not what you thought, then you'll get a whole lot of learning involved, and this will involve most of the neurons of your cortex, which are in fact layer 2-3 neurons. But it will boil down to some congruency somewhere in the layer 5 neurons, and for the least energy, you'll have the congruence coming from the column. So this would be the claim that the output of the column is in fact a congruent, let's say, expectation, but it's generated and it's regulated in terms of learning and plasticity by incongruence. And so you're devoting, actually, in the end, most of your energy to the incongruence, but you're using the minimum of energy to actually do something about it. That would be my answer in a complex way.
Hi. I think my question perhaps changed a bit now because of this last part of the conversation, but I'm going to go back a bit. Then, about the layer 6B, would you predict that in a freely moving awake animal, there is a lot of activity of the layer 6B at constant in a sense of searching the space, which you mentioned before?
Yeah, I would predict that, yeah.
And then when would it be then inactivated, by who? Because that I felt like I was missing a bit, like where is the source of inhibition for layer 6B? And if that comes, it should certainly not inhibit the entirety of it, right?
Yeah. I left out an important statement, which was that the transgenic animals recruit half of the excitatory neurons, or at least half of the neurons of 6B, which are excitatory, and the other half are inhibitory. So there are local inhibitory neurons in 6B. And the other thing that I left out was that apart from its very specific input to layer 5 neurons, both at the top and in the middle, it also hits interneurons, but only in layer 6A. So there is some inhibition both within 6B and of 6A. And this will be important because 6A is also part of the thalamocortical loop. So eventually there's an inhibitory story that I've completely left out at this point, just because we don't have the wherewithal to investigate it.
Hi, my name is Bjørnar Hassel. I'm a clinical neurologist, and I work with patients who are troubled with fatigue after brain infections and other insults. My question is really, can you somehow shut off or turn down layer 6B? And I'll give you the reason for my question. We see that in patients who are troubled by fatigue and who don't have the wakefulness and the attention that you ascribe to layer 6B, they have a reduced activation of much of their left hemisphere and of their left collicular superior. The impression that I get from studying these patients is that somehow there is a switch that is turned on the time of that they suffer from their lesion, brain lesion, and it's not, and it stays turned on, this fatigue response. And I wonder, so here part of my question is, do you know of any mechanism that would turn down the 6B function? The other thing, is it lateralized somehow? We see this.
That's a good question. It ought to be lateralized.
Turning on the left side.
Oh, you mean is that mechanism stronger on one side than the other? This I couldn't go, and I'm really out of my depth here to. So there are many things, many ways to regulate 6B, unfortunately, because I don't know exactly where to start. Trying to predict for any particular disorder that would relate to 6B what the starting point would be. So there are too many reasons that one could speculate 6B are affected. But so what I think, armed with all of this knowledge, you should, I think, go to the specifics of whatever drugs that you know affect this and see which receptors they are. I almost can guarantee at this point that 6B has these receptors, but I would first caution to look at that. And unfortunately, as usual, you'll find that these receptors are not just in 6B, but there will be undoubtedly a 6B story, a possible 6B story. Then the question becomes, how do you investigate this? So if I'm saying nothing else, it's today. Adding 6B gives you another place to look, and a really important place to look. None of my predictions are so firm that I can just standing here answer what was presumably a complex question. But I'm just saying that if you ignore 6B, you do it at your own peril. Now, you know, it's all too likely that 6B would be involved in such a disorder, but the details are going to matter here.
Thank you.
Last question.
Yes, thanks for a very interesting talk and a lot of information. I thought it was very interesting that you had this short-term facilitation from layer 6B and that it also lived on for some time. Yeah. And I was just wondering, how do you, if you want to get back to your previous working memory sort of immediately, how do you get the specificity? I guess it's not sort of Hebbian, or because it's this plateau potential is in the apical tuft, right? So it doesn't. So where is the specificity? Yes. And then you also said that, for instance, layer 6B don't target layer 2, 3, and those are supposed to increase the gain in layer 5 and be involved in working memory. Did you have some big picture?
I have some thoughts, but we're missing the big picture here. But the first thing I want to say is that the thing that most recruits layer 6B, and another thing I failed to mention, are the IT neurons, not the ET neurons. So it's the IT neurons of the cortex that are having the biggest effect, both layer 2, 3 and layer 5A, but particularly layer 5A, because these are having also long-range connectivity, and particularly layer 5A coming down from prefrontal cortex. And so one could imagine a cascade of events, because the layer 2, 3 activate the layer 5A as well. So you have I won't go too much into what else I think is true of the IT neurons, but basically, to a first approximation, these are not the congruent contextualized neurons that are part of your expectation, as we were just discussing, but are doing nitty-gritty kinds of operations within the cortical column that leads to, in the end, congruence or not. And now when we're talking about these are feeding eventually into layer 6B, and layer 6B is now going and has the plasticity that you're just referring to. So I guess that as you use your brain, your cortex, you're by hook or by crook, either stumbling across or directing yourself to or being influenced by the outside world, such that you recruit here and there the IT neurons. Were they to be part of a loop that made you aware of something, then there's a trace of that in the connectivity of layer 6B that captured that loop for you. And it's going to survive the thought itself. So if you stumble into that mode again, you'll be that much quicker to recruit the loop through these facilitated synapses. That's where I have to stop there, not just because we've run out of time, but because I don't have anything more to say than that.